Good evening, everybody. How are you, Dr. Abbott?
Good. How are you doing?
I'm good. Where are you joining us from tonight?
Denver, Colorado.
Okay, so we're here on the East Coast, so I always say good evening to everybody. Well, it's still evening for everybody, but welcome to tonight's Woo You event, Pharmacologic Treatments for Presbyopia, Navigating Current Options with Dr. Caleb Abbott. Thank you. I'm your host, Dr. Jennifer Stewart, and thank you to Lens Therapeutics for exhibiting at tonight's event. Just some reminders for housekeeping. For each hour of CE, attendees must be online for a minimum of 50 minutes. For a COPE certificate, please fill out the survey link in the chat. The survey link will also appear when the webinar ends. CE certificates will be delivered by email and sent to ARBO with OE tracker numbers. We'll also display a QR code at the end of the event if you have the OE tracker app on your phone. CE certificates will be emailed within 4 weeks, and feel free to ask questions throughout the presentation using chat or Q&A. If you're on your phone, here's where you can find both of those. And it is my great pleasure tonight to welcome Dr. Caleb Abbott to Woo You. He's an optometrist and assistant professor of ophthalmology at the University of Colorado School of Medicine. He's affiliated with both the Dry Eye Clinic and the Center for Ocular Inflammation. In addition to the clinical and research roles, he serves as vice president for the Ocular Wellness and Nutrition Society, is chair of the Nutrition, Disease Prevention, and Wellness Special Interest Group for the American Academy of Optometry, and is a member of the Advisory Council for the Academic Medical Center Optometry AAO SIG. He also holds a position on the editorial advisor board for Modern Optometry and Optometry 360 and is a graduate of the AAO FLOM Leadership Academy. He also hosts Dry Eye and Ocular Surface Disease section of the clinical podcast series through the American Academy of Optometry Foundation. In 2024, he was nominated for Colorado's Young Optometrist of the Year and recognized as a one to watch by Modern Optometry. In 2019, he co-founded SunSnap Kids, a startup that won first place in the inaugural Bright Ideas Pitch Competition in 2022 and third place in the Optometry Innovation Awards in 2023. He recently sold the majority of the company to focus more on his clinical and research responsibilities. When he's not seeing patients, conducting research, or working on SunSnap Kids, he lectures on ocular surface disease, writes articles, and serves as a medical reviewer for multiple journals, including the Ocular Surface and Optometry and Vision Science. He resides in Denver, Colorado with his wife, daughter, and newborn twins. Congratulations.
Thank you. I was not expecting you to read all of that, but
I know
thank you very
it's
much.
hard to leave stuff out on the fly, and it's so hard to pick and choose because there's so much great stuff. So you should have your due, and we're very excited to have you here. So I'm going to turn it over to you. I will be back at the end. Reminder, put those questions in the chat and Q&A, and we will have time. If we have time at the end, we'll get through as many as we can. I'll turn it over to you.
All right, that sounds good. Well, these are my financial disclosures, all of which have been mitigated for this talk. So tonight we're going to be talking about presbyopia. So for those of y'all that are listening, I want you to just think, how many of y'all actually have presbyopia? And for those of y'all that do not have presbyopia, how many of y'all will eventually become presbyopic? And for those of y'all that claim to be immune to presbyopia, how many of you have a problem with lying? So the fact is that everybody either has or gets presbyopia if you live long enough. And I want you to think, can you think of any other ocular condition that 100% of your patients are either currently have or are guaranteed to develop in the future? Because there's really not a whole lot of them that 100% of patients will eventually get. So presbyopia is inevitable. What else in life is inevitable? I listed some things here that I could think of that are inevitable. So death is inevitable. Taxes, unfortunately, are inevitable. Sarcopenia, or the gradual wasting away of our muscles as we get older, is definitely inevitable. Cataracts are inevitable if you live long enough. You may not need surgery, but you will definitely have cataracts if you live long enough. And skin aging is inevitable. And when I try to think about all the different things in life that are truly inevitable, those are really the only things that I came up with. So presbyopia, in addition to these, if you come up with anything else that you feel like is inevitable, you should drop it in the chat. But there's not a lot in life that we know that we are guaranteed to get in life, or that is guaranteed to happen to us. So everybody eventually gets presbyopia. We know that about 1.8 billion people worldwide have presbyopia, and 130 million Americans have presbyopia. This constitutes about one-third of the US population. So many of our patients are coming in and they're complaining of the inability to see up close whenever they are corrected for that distance vision. This is something that you and I are dealing with all day, every day in our patient population. It's not new to us, but it is new to a lot of our patients. So aside from just the blurry vision, patients end up being very, very affected by presbyopia. It really does affect their quality of life. So there's a lot of patients that actually are embarrassed of growing older. And one of the first signs of growing older is you start to get that long arm syndrome. You're holding things out further away, trying to make it clear because you've lost that ability to accommodate up close. Patients actually get anxious about this. They get embarrassed about this, and it reminds them that life is short, that they're aging. There are studies that show us that presbyopia, the onset of it, results in diminished self-esteem, that it affects our social interactions. If you go out and you forget your reading glasses or your contact lens prescription isn't right and you can't see up close very well, it kind of limits what you're doing. You might have a hard time reading the menu at a restaurant or checking your cell phone for text messages. And it really does alter day-to-day activities. Those of y'all that have presbyopia, I think know this. It really is a kind of game changer and a big effect on quality of life when this kicks in. And now this is something that you're dealing with really all day, every day, and trying to figure out ways to work around it. So what is happening here? So with our normal distance vision, whenever we're younger, we have a relaxed ciliary muscle, and that is focusing our distance vision far away. And then when we are not presbyopic and we need to look up close, we contract the ciliary muscle. And when we contract the ciliary muscle, it's going to focus the light onto the retina, which is going to let us see up close. And it is during this time that we get what we learned about in optometry school called the near triad. In the near triad, we have accommodation, convergence, so our eyes are going to converge to focus on what's up close, and then meiosis. We actually get a meiosis effect that is trying to increase our depth of focus. And when we go to look at something up close, all three of these things happen: accommodation, convergence, and meiosis. But what's happening in presbyopia is when that ciliary muscle contracts, you have insufficient accommodation and you're no longer actually able to focus up close any longer. And that's when we say that you or that patient has become presbyopic. And these are the options that we really have for presbyopia right now, or the historical options. We have glasses. So reading glasses, single vision near. We've got multifocal lenses, bifocals, trifocals, progressive lenses. For contact lenses, we've got distance vision, overspect. We've got monovision. We have different types of multifocal contact lenses. And then we've tried to come up with surgical options to help these patients out too. So things like corneal inlays that is trying to create a miotic effect, and you're looking through a small pinhole. There's things like intraocular lenses that are multifocal that are trying to focus up close as well. But really, a lot of patients don't like a lot of these options. And there's an unmet need for a pharmacologic option that we see here. And when I say unmet need, what I'm really talking about is that patients oftentimes don't want glasses. They're not satisfied with contact lenses, multifocal lenses. They're not satisfied with the multifocal intraocular lenses. What they really want is a drop that is going to help them out. And up until recently, we had no options for this. And really the focus of tonight's talk is going to be going into what are the options that we now have for patients to help address this unmet need, and then what are options that are also coming in the future. So if we were to create a drop that is going to improve near vision, how would we do it? Well, the first way that we could go about this is trying to restore the natural flexibility of our crystalline lens. This is easier said than done. It's actually quite difficult to do this. We have some researchers that are in our institution that are actually working on trying to come up with this. But we are quite a bit a ways away from having a drop that can successfully do this, probably at least 10 years. Modifying scleral biomechanics. This one is not intuitive, but if we actually make the sclera more flexible, it's going to assist our ciliary muscle at actually contracting and changing the shape of our lens. So if we modify scleral biomechanics, this could potentially improve the near vision. We do not have any drops that can do that, but we will talk in a little bit about one surgery that is trying to do that. And really, the option that we have right now is pupillary constriction, also known as meiosis. This is the best option that we have at the moment to treat presbyopia in our patients. So the goal of meiosis is to increase depth of focus, but without significantly compromising distance visual acuity. due to ciliary muscle stimulation. The real problem is that most drops that stimulate, that cause meiosis, they also contract the ciliary muscle. When you contract the ciliary muscle, you get this anterior lens displacement. When the lens moves anteriorly, you then get a myopic shift. So that will improve the near vision. But if you get too much of a myopic shift, that is going to be at the detriment of the distance vision. So this is why when it comes to FDA clinical trials, what they're really looking at, the FDA that is can we improve near vision without sacrificing a line of distance visual acuity? So when it comes to increasing depth of focus through meiosis, pupil size really matters. So what we have here in this graph on the y-axis is total depth of focus. So the additional depth of focus that we're getting with pupillary constriction. On the x-axis is pupil diameter. What we see is that as your pupil goes down and down in size from 6 millimeters to 5 to 4 to 3, you start to see a slight increase in depth of focus, but not a lot. It is kind of right here when you get to that 2 millimeters pupil that you start to see a little bit of an increase in that depth of focus. But once you get to that sub 2 millimeter pupil, so right here is about 1.75, here's probably about 1.5, 1.25. This is when you see a dramatic increase in the total additional depth of focus that your patients are going to be getting. So if we can get to this kind of sub 2 millimeter pupil, that's when we're starting to get a big improvement in our increased depth of focus. This is actually looking at the pupil diameter on the x-axis, the same as the last, but instead on the y-axis, we're actually looking at lines of improvement. So how many lines of near vision improvement you get. And then in the different shades of color here, we have different lighting conditions. And again, what we see is that 2 millimeters is really that inflection point. 2 millimeters is really the difference between when you start to get a little bit of increased lines of near vision improvement. But once you get to that sub 2 millimeter mark, you start to see that there's a skyrocketing effect of a lot more increased lines of near vision improvement. And this is true regardless of the lighting condition. This is true in bright lights. This is true in kind of mesopic conditions and also dark lights as well. But how small is too small? And this has generally been the question that a lot of people have been asking. We want to constrict the pupil. We know that that's going to improve the depth of focus, that this is going to improve the near vision, but can you constrict the pupil so much that it actually starts to cause problems for our patients? Well, it looks like you can definitely get sub 2 millimeters without significantly impairing perceived brightness. So that much we know is true. There's a study that looked at what point of pupillary constriction do you start to lose some of that peripheral vision. If you constrict the pupil a lot, conceivably, you're now losing some peripheral vision. And they found that a 1.5 millimeter aperture did not result in any visual field loss. And then what about diffractive blur, which we'll talk about more towards the end of the presentation. But, there's studies that differ on this, but it seems that diffractive blur definitely becomes a concern when the pupil goes down to 1 millimeter and below. Probably really doesn't start to become a concern until about maybe 1.3 millimeters or so. But definitely at 1 millimeter, that's when we start to get this effect where the light is trying to bend around the pupil. When it's bending around the pupil, it starts to induce some additional blur. But that doesn't seem to be a problem until definitely at 1 millimeter, but probably not really till 1.3 millimeters. So these are the pharmaceutical treatment options that we have here. So we can either have a cholinergic muscarinic agent, so something that is stimulating that cholinergic muscarinic receptor functioning as an agonist. This will induce a strong miotic effect on the iris sphincter. So these drugs work by targeting the iris sphincter, stimulating that iris sphincter, causing pupillary constriction. There are two different types of drugs that we have here. The first is non-selective. By non-selective, it means that it is going to stimulate the iris sphincter, but it's also going to stimulate the ciliary muscle. So it is not really selective at what it's trying to target. It is stimulating iris sphincter and ciliary muscle. But then we have selective cholinergic muscarinic agents that actually have a minimal effect on the ciliary muscle that are really precisely targeting just that iris sphincter. So those are the options that we have when it comes to cholinergic muscarinic agents. But then we also have sympathomimetic agents. So this is working through the sympathetic nervous system, but it's working through very specific channels. And what it's trying to do is actually inhibit the iris dilator muscle. So if you inhibit something that dilates, the effect that you're going to have is pupillary constriction because you're inhibiting the very thing that's trying to hold the pupil in a larger size. When you inhibit that, the pupil is going to constrict. This has a weak miotic effect, so not as strong as a miotic effect, is actually directly stimulating the iris sphincter. But one benefit is that it does not stimulate the ciliary muscle, so you don't start to get some complications associated with ciliary muscle stimulation. So here are the problems that can occur if you end up stimulating that ciliary muscle. The first one we've talked about a little bit. This is myopic shift or pseudomyopia. If you stimulate that ciliary muscle, you get a little bit of this anterior lens displacement. This will improve near vision when you get the myopic shift because you're making yourself a little bit more nearsighted, but it has the effect of actually worsening distance vision. If you end up inducing too much of a myopic shift, all of a sudden now your distance vision is blurry. Another problem with stimulating the ciliary muscle is that you can get a ciliary spasm headache. This is something that we know that happens with certain agents that stimulate the ciliary muscle. It's a known problem. And so patients can get headaches associated with this. When we also get this anterior lens displacement, this is when all of a sudden we start talking about traction. This is what everybody talks about when it comes to miotic drops. What sort of traction effect is it happening? So when you get that anterior lens displacement, you do get some vitreoretinal traction. This can lead to retinal tears and retinal detachments. If that lens moves too far anteriorly, now that anterior lens capsule can start touching the posterior iris. If that anterior lens capsule touches the posterior iris, you can start to get posterior synechiae formation. I actually had a patient that had this last week, but it was not due to a miotic drop. It was actually due to him having scleritis and getting a lot of uveitis associated with that. So when it comes to selective versus non-selective cholinergic agonists, a non-selective cholinergic agonist, again, is stimulating the ciliary muscle in addition to the iris sphincter. We want the iris sphincter to be stimulated. We do not want the ciliary muscle to be stimulated. When it comes to a selective cholinergic agonist, this is selectively targeting the iris sphincter while actually sparing the ciliary muscle. And when you do that, it mitigates complications that are associated with ciliary muscle stimulation, like myopic shift, ciliary spasms, and retinal traction. So this is an example of just two different ingredients that we have in the difference between being selective and non-selective. So up at top here, we have aceclidine, and at the bottom, we have pilocarpine. Most of us are familiar with pilocarpine. Most of us are probably not familiar with aceclidine. It is actually a new compound in the United States. It's never been used before until it's actually going to start to be used next month. So what we see here in kind of this middle section is the amount of drug required to elicit a 50% of the maximum muscle response. So essentially, how much drug do you need to stimulate that particular muscle? So when we look at pilocarpine, we'll look at this first. What we see is that it needs a little bit of, 1,800 to stimulate the iris sphincter. It needs about 2,800 or 3,300 to start stimulating the ciliary muscle. So it needs a little bit more to stimulate the ciliary muscle. This makes it to where it is selective for the iris at about a 2-to-1 ratio. So selective for the iris twice as much as the ciliary muscle. If we look at other compounds that are more selective, though, for the iris sphincter, we can look at aceclidine. Aceclidine, it takes here 900 nanomoles per liter to actually stimulate the iris sphincter muscle, but it takes 20 to 25,000 to actually stimulate the ciliary muscle. So what this means in a gist is that it's much more selective for the iris. So it's about a ratio of about 20-to-1 in terms of how selective it is for the iris as opposed to the ciliary muscle. And we want something to be, again, more selective to the iris and not be stimulating the ciliary muscle as much. It takes way more drug for aceclidine to stimulate the ciliary muscle than it takes for pilocarpine to stimulate the ciliary muscle. It doesn't take nearly as much of the drug for pilocarpine to stimulate that ciliary muscle. So how does this translate into side effects? So if we were to take 2% aceclidine and we were to take 2% pilocarpine, we would see that they would have similar iris sphincter stimulation. This is leading to a pupil size of 1.4 versus 1.35, so pretty similar. But if we look at the corresponding ciliary stimulation as measured through myopic shift, what we'd see is that 2% pilocarpine is going to yield a minus 1.3 diopter myopic shift. So a huge myopic shift, but that same concentration of aceclidine is only going to yield minus 0.17 diopters of myopic shift. So what this is really telling us is that aceclidine is not stimulating the ciliary muscle very much, whereas pilocarpine is stimulating the ciliary muscle quite a bit. And again, so that is the difference between something being selective towards the iris sphincter versus not selective for the iris sphincter. So when it comes to these cholinergic agonists, whether they're selective or non-selective, what it's trying to do is constrict the pupil by stimulating the iris sphincter, give a strong miotic effect. But again, the cons are that if it is a non-selective ingredient, it can then stimulate that ciliary muscle. So these are the ingredients that we have that are either available to us now or will be coming out in the future. So we have two pilocarpine-based options. These are the non-selective ones. So they do stimulate the ciliary muscle. It does not spare the ciliary muscle. These drugs are Vuity and Colosi. And then we have an ingredient called carbachol. This is also a non-selective cholinergic muscarinic agonist. It does not spare the ciliary muscle, so it is non-selective in that sense. And the PDUFA date, or the FDA decision date for that drug, is January 28th of next year. And then we have aceclidine. This is the only one that is a selective cholinergic muscarinic agonist. It does spare that ciliary muscle, and it was just FDA approved in July of this year and should be commercially available really any day now at this point, probably next month though. And then we have sympathomimetic agents. So the method of action for these, again, is indirectly causing meiosis by reducing dilator activity. The pros is that there's no ciliary muscle stimulation. The cons is that there's less miotic effect compared to those cholinergics. It's unable alone to achieve a sub 2 millimeter pupil. So examples of this is brimonidine, which we're all familiar with. It does spare the ciliary muscle. The PDUFA date for that is January 28th, 2026. And you might say, wait, I just saw that PDUFA date on the other slide. Yes, that is because carbachol is actually going to be combined with brimonidine into a single drop that is going to be used for presbyopia. And that's why both of those have the same PDUFA date, because it's actually going to be part of the same drug. And then there's phentolamine, which is not really being investigated for presbyopia, but mostly being investigated to reduce halos and glare post-refractive surgery at nighttime. That one does not have a PDUFA date yet. It's still in FDA clinical trials. So these are overall the pharmaceutical presbyopia options that we either have now or that are coming to the market pretty soon. So we have Vuity, which is pilocarpine 1.25%. This was approved in 2021. We have Colosi, pilocarpine 0.4%. This was approved in 2023. We have Viz, or aceclidine, 1.44%. That was approved 2 months ago, I guess, so July 2025. And then we have Brimacol, which is that combo drop with carbachol and brimonidine that has finished their phase 3 clinical trials, and the PDUFA date is January of next year. So we're going to go through kind of each of these drugs, the pros and cons of each of the drugs, and then go over some future options that we're going to have kind of towards the end of this presentation. But first, before we go into each of these drugs, you have to understand the FDA primary and secondary endpoints. So the FDA actually determines what the primary endpoint is. What the FDA wants to see with these drugs is the percentage of patients that have a 3-line or greater improvement in distance corrected near vision acuity. They want to see it under mesopic illumination, and they want to see that compared to the control. They also want to see that there is no loss of distance corrected acuity that is more than 1 line. So essentially, they just want to know the percentage of patients that end up achieving that 3 lines or greater improvement in near vision without losing any distance vision visual acuity. The FDA allows it to either be monocular or binocular. It does not specify which one it has to be. So we'll kind of point out in the clinical trials which one each drug ended up using, because that does make a big difference if you're looking with one eye or looking with both eyes. Secondary endpoints are determined by the company, not set by the FDA. So a little bit of history here. So we'll start with the history of pilocarpine. Pilocarpine was actually isolated from the leaves of the Pilocarpus species of plants found in Brazil way back in 1874. In the late 19th century, it was actually introduced into a medicine as a treatment for glaucoma. So it's been used to treat glaucoma for a very long time. It was the first drop used to treat glaucoma. It is trying to reduce intraocular pressure by causing miosis or pupil constriction and increasing aqueous humor outflow. Fast forward to the 20th century, started looking at applications for treating xerostomia, which is dry mouth, for Sjogren's syndrome, or for patients undergoing radiation therapy to the head and to the neck. Fast forward to the 1990s and 2000s, it actually was approved for systemic use in a drug called Saligen to stimulate salivary secretions. Now, most of my job includes treating severe dry eye patients, and I actually prescribe oral Saligen in my Sjogren's patients to boost not only the salivary secretions, but to boost lacrimal gland secretions as well. And oral pilocarpine does work very well for that. So the first drug we'll talk about is Vuity. So Vuity was FDA approved in 2021. This is 1.25% pilocarpine. It is preserved with BAK. It is indicated for the treatment of presbyopia in adults, and the dosing is twice a day. So pay attention to the dosing throughout the presentation. Some of these drops you can dose twice a day. Some of these drops you only actually have to dose once a day. And Vuity, being pilocarpine based, is a non-selective cholinergic muscarinic agonist, so it is going to target the iris sphincter, but also have effects on the ciliary muscle. The population that was studied with Vuity was ages 40 to 55. It was studied from -4 diopters to +1 diopters, spherical equivalent. It was not studied in patients that had had cataract surgery, so no pseudophakes, and the binocular vision is what was reported. So in the Gemini study, 31% of participants on Vuity ended up achieving 3 lines or greater of near vision improvement, so almost a third of patients. In the Gemini 2 study, we see 26% of patients achieved that primary endpoint of 3 lines or greater of near vision improvement. And then in the Virgo studies, at hour 9 versus the placebo, 35% of patients. So really, it's anywhere from about a quarter to about a third of patients with Vuity achieved that 3 lines or greater improvement in near vision acuity. Now we're going to move on to Qlosi. So Qlosi was FDA approved in 2023. This is pilocarpine, 0.4%. One benefit of Qlosi is that it is preservative free. So it has a near neutral pH to improve corneal penetration, which is why you can kind of get away with having a lower concentration of pilocarpine. And if you want it to last all day, you really have to use Qlosi twice a day. If you use it once a day, it's probably going to last 4 to 6 hours, but it's not going to last the entire day. So if you want it to last all day, you have to do BID dosing. And it is a low dose formulation, which is supposed to result in less side effects as compared to Vuity, which is a higher concentration pilocarpine. And this is a non-selective cholinergic muscarinic agonist, so hitting the iris sphincter and the ciliary muscle. Qlosi was studied in a little bit broader of a population, so 45 years of age to 64 years of age. So studied really from early presbyopes to patients that are really getting near that absolute presbyopia. It was studied in patients that had pseudophakia as well as patients with their natural lens. And it is actually studied for monocular visual acuity. That's what was reported as compared to Vuity, which reported binocular vision. So the primary endpoint, again, 3 or more lines of near vision improvement. In the near 1 and near 2 studies, we see 39% of patients ended up having meeting that FDA endpoint of 3 lines or greater of near vision improvement at hour 1 post dose 1 on day 8, and then 48% post dose 2 on day 8. The maximum response that we see with Qlosi is 55%. So at best, what we see is that 55% of patients had 3 lines or more of near vision improvement. That was after the second dose on day 15. So what I want to do here, I can really only present as much data as has been released by the companies. Qlosi has released more data, therefore I can present more data on it. So at day 1, we're going to look at how they did at day 1. So at day 1, 30 minutes in, 16.8% of patients had 3 lines or greater improvement in near vision acuity. So about 17% of patients had met that. That was when the pupil had constricted down to 2.5 millimeters. So 2.5 millimeters is not particularly small, so you're starting to get a little bit of that miotic effect, but not enough to have a large percentage of patients achieve that 3 lines or greater of near vision improvement. If we fast forward on day one to the 3 hour mark, this is when it was the most effective. It was the most effective on day one when 36.7% of patients achieved 3 lines or greater of near vision improvement. And that corresponds to when the pupil had constricted down to 2.1 millimeters. And then if we look at their day 15 data, so they hit their maximum response. So the best that the drug worked was on day 15 at the 4 hour mark after that second dose. We see that 55% of patients had 3 lines or greater of near vision improvement. And again, this was done monocularly. That was when the people had constricted down to 2.2. So Beauty had really gotten, or Calosi had really gotten down to about 2.1, 2.2 millimeters of pupil size. So when we look at pilocarpines in general, though, this is kind of an effect of when you're trying to not have side effects due to stimulating the ciliary muscle. You have to have a balance between how the concentration that you give. If you give too much concentration, there's more side effects where you get more of a miotic effect. If you don't give enough of a concentration, you don't have as much of a miotic effect. You won't have many side effects, but you won't have as much of a miotic effect. In general, what we see with pilocarpine based drops, this is a systematic review. You get down to pupil sizes like 2.5, 2.5 here, 2.4, 2.4. So you're really getting into that mid 2 millimeter pupil size range. Now we're going to fast forward to aceclidine. So aceclidine was synthesized in the 1950s, 1960s. It was originally supposed to be an alternative to pilocarpine for glaucoma. It didn't really turn into that. It was never really used as much as pilocarpine to treat glaucoma, although it still was used to treat glaucoma, but only in Europe. In the mid 20th century, it became that glaucoma adjunct therapy. It induces miosis, lowers IOP by enhancing aqueous humor outflow. So from the 2000s to the present, it's really no longer used for glaucoma. There's more effective drops, beta blockers, CAIs, prostaglandin analogs, but it was used for glaucoma in Europe until roughly 2010 or 2013, somewhere around there. Now, aceclidine is a pupil selective cholinergic biotic. It has a high affinity for the iris sphincter and minimal impact on the ciliary body because it is a selective cholinergic biotic. It is able to achieve a sub 2 millimeter pupil size, and it does have extended history of safety based on Europeans using it for glaucoma. So actually, it has been dosed at least 400 million times without a single report of a retinal detachment or retinal tear. If you try to search aceclidine and find if there's a connection of aceclidine to retinal detachment, you can't find it, or at least I can't find it whenever I search all through PubMed and Google Scholar. So the drug that we're going to be talking about with the aceclidine is Viz. So Viz was FDA approved July of 2025. This is aceclidine, 1.44%. It is preservative free. And this is the first drop that we're talking about that has only daily dosing. So the two pilocarpine based options, if you want them to last all day, you really have to use them twice a day. But this is the first drop that just has daily dosing. So it is a selective cholinergic muscarinic agonist, high affinity for the iris sphincter with minimal impact on the ciliary body. It was studied in ages 45 to 75 from -4 to +1 diopters in pseudophakic as well as phakic patients. It was also studied in post-LASIK and PRK patients, which is something new. And monocular visual acuity was reported. So again, the primary endpoint is what percentage of patients had 3 or more lines of near vision improvement. What we see in the Clarity 1 and Clarity 2 studies, when you pull it together, is that at 1 hour post, 1 hour post the only dose on day one, 73% of patients had met that primary endpoint. And at the 3 hour mark on day one, 71% of patients had met that primary endpoint. So this is a graph that is showing the percentage of participants achieving a 3 or more lines of near vision improvement. This is on the y-axis, and then on the x-axis, we have time. So we see at 30 minutes out, 71% of patients are having 3 lines or more of near vision improvement. At 1 hour, 73. At 3 hours, 71. And then it slowly starts to go down. So 62% at 5 hours, 57 at 7 hours, 48 at 8 hours, 49 at 9 hours. And it was only measured until the 10-hour mark. But at the 10-hour mark, 40% of patients still achieved a 3-line or greater improvement in near vision acuity. If you were to trace this line and kind of see what direction it's going to go, you'd probably see that if they did test it at 11 hours, you'd probably still have some patients in the 30s. You test at 12 hours, who knows? But the study actually stopped at that 10-hour mark, so that is all the data that we actually have. But conceivably, it probably lasts a little bit longer than that. This is a secondary endpoint. This is the percentage of patients that achieved a 2-line or greater improvement in near vision. What we see, 90% of patients, like 91 to 95% at the beginning had that, but still 69% of patients at the 10-hour mark still had 2 lines or greater of near vision improvement. What is really interesting about aceclidine is this chart here. So this is the percentage of patients that achieved 1 line or greater of distance visual acuity improvement. So not near, we're talking distance vision at this point. Now remember, with the non-selective cholinergic agonists, you're probably going to worsen distance vision a little bit, or you're hoping that you're coming up with a concentration that's not going to worsen distance vision. But with aceclidine, it does not stimulate that ciliary muscle. Therefore, you're getting a true pinhole effect without inducing that myopic shift. So when you do that, you're actually getting a pinhole effect that can improve your distance vision. So interestingly, we see at maximal effect, this is 5 hours after using the drop, that 41% of patients actually had a 1 or greater line improvement in distance visual acuity. So I thought that was pretty interesting. Really, at the 10-hour mark, 31% of patients had that. So somewhere around 30 to 41% of patients will have a little bit improvement in their distance visual acuity, which is really interesting. And again, I'm able to show the data only that has been released by the companies. So Viz or Lens Therapeutics has released quite a bit of data, which is why I'm able to go a little bit more into this. So this is a graph showing the percentage probability of achieving 3 lines of near vision improvement by pupil size. And I think this is important to understand how these miotics work. So let's say a patient had a pupil constriction that's anywhere from 2 to 2.5. What we're looking at are these patients here. They would have between a 25 and 38% chance of achieving 3 lines or greater of near vision improvement. When you get down to 1.75 to 2 millimeter pupil, about half of those patients were going to meet that FDA endpoint. When you're from 1.5 to 1.75 millimeters, about 68% of patients, that was the probability of having 3 or more lines of improvement of near vision acuity. When you got to less than 1.5 millimeter pupil, that's when 78%, there's a 78% probability of achieving 3 lines or greater of that near vision improvement. So what we see is that really when you get to roughly that 2 millimeter pupil and lower, that's when we start to see, again, unsurprisingly, based off some of the data we saw earlier, that's when you start to see that you get a lot more improvement in that near vision. All right, now we're going to move on to the last 2 ingredients, which are carbachol and brimonidine. So carbachol was synthesized in the 1930s as a long-acting analog of acetylcholine. It resists acetylcholinesterase breakdown. This causes sustained parasympathomimetic effects. From the 1950s to 1972, it was used as adjunct glaucoma therapy for similar reasons to the other drops that we discussed. But it wasn't actually used a whole lot topically. So it was difficult to get it to absorb through the cornea. There were some side effects, namely posterior synechia, happened quite a bit with carbachol used topically. So from the 1970s onward, it wasn't really used at all as a glaucoma drop. It actually found a niche for use during intraocular surgery, specifically during cataract surgery for a rapid, reliable meiosis. And then we have brimonidine. Brimonidine synthesized in the 1990s as a highly selective alpha-2 adrenergic agonist to treat glaucoma and ocular hypertension. 1996, it received FDA approval to be a glaucoma drop. It works by decreasing aqueous humor production and enhancing uveoscleral outflow. In the 2000s, it became widely adopted for use in glaucoma. It's also believed that it could have some neuroprotective effects, which is a benefit of brimonidine. In present day, I think all of us probably have used brimonidine in patients. We use it quite a bit for glaucoma management. We also all know that there's an over-the-counter drop that is a weak version of brimonidine that is used to treat ocular redness. Because brimonidine is a vasoconstrictor, it can be used off-label or in the over-the-counter option to actually decrease redness of the eye. So Brimacol is the last drop that we're talking about, and this is a combo drop. So the PDUFA date for this drop is January 28th of next year. This is carbachol, a non-selective cholinergic agent, mixed with brimonidine, which is that alpha-2 agonist. So carbachol is going to stimulate the iris sphincter. It's also going to stimulate the ciliary muscle, but brimonidine is only going to inhibit that iris dilator. So indirectly causing meiosis by inhibiting something that dilates. This drop is also only daily dosing, which is really nice. So the first two drops we talked about really are BID dosing. The second two drops we're talking about are daily dosing, which is a nice benefit for patients. And this one is also preservative free. So according to the company's website, the benefits of adding brimonidine is that it may inhibit contraction of the ciliary muscle by activating alpha-2 receptors, potentially mitigating the side effects associated with miotics, which is very interesting. It appears to increase the bioavailability of carbachol by altering aqueous dynamics, prolonging the pinhole effect and leading to a longer duration of action. And then you also have the benefit again of inhibiting that iris dilator muscle. So that is why brimonidine was combined with carbachol in this particular drop. So we'll look at the data that's been released for this drop. So this was studied in the ages 45 to 80. It's actually a very, very broad range of patients were studied. But it was only studied in emetropic patients. I do not know why it was only studied in emetropic patients. All of the other drops seem to be studied mostly from about -4 to about +1 or so. But this drop was only studied in emetropic patients. It was studied in pseudophakic as well as phakic patients, and binocular vision was reported. So not monocular, but binocular. The primary endpoints, again, 3 lines or greater of near vision improvement. All we know is that in the BRIO 1 and 2 studies, they achieved their primary endpoint at all time points, up to 8 hours. We know that it improved reading speed and patient-reported quality of life outcomes, and it improved conjunctival hyperemia. But unfortunately, the specific data for this drop has not been released by the company. So there's nobody that really knows the specific data except people in the company and the FDA at this point, if it's been submitted to the FDA, which it has. So what do we know? We know that it was studied in emmetropes only. We know it's once daily dosing, which is a nice benefit. We know it lasts up to 8 hours, so a pretty good duration of action. We know it met its primary endpoints at every single hour, assessed with binocular vision. And we know through the company releasing a press release that it improved reading speeds, quality of life, and redness through the brimonidine being in the drop. But unfortunately, that's really all that we know here. So these are the pupil size outcomes with presbyopic miotic agents. With Vuity, we see really a range of maybe 2.3 to 2.5 millimeters. With Colosi, we see a range of 2.1 to 2.5. With Viz, we see that 99.6% of people achieved a sub 2 millimeter pupil. The mean minimum seems to be at 1.54 millimeters. So a lot more pupillary constriction. With Brimacol, we know that the range seems to be 2 to 2.5, but I think that number is probably a little bit closer to 2. So probably a little bit more constriction with Brimacol than with those pilocarpine-based options. So this is a table that I made that I tried to summarize really all the data that is publicly available for all 4 of these drops. So we have the dosing here. So we see the pilocarpine-based options. If you want it to last all day, it's really twice a day. So 1 drop usually lasts 6 hours or 4 hours. With aceclidine, it's 1 drop a day. With Brimacol, it's 1 drop a day. We can see the responder rate at 1 hour and at 3 hours. We see that with the pilocarpine-based options. For day 30 with Vuity, it was about 26 to 31% of patients were responders. With Colosi, it was about 37% of patients at day 1 had responded. With Viz, it was 71% of patients. And then with Brimacol, that has not been reported. We can see the testing method. So binocular for Vuity and Brimacol, monocular for Colosi and Viz. We can look at the populations that were studied. We can look at pupil size. We can see if it's preservative free. All of them are preservative free except Vuity, which is preserved with BAK. And then we can see which ones spare the ciliary muscle. All of them stimulate the ciliary muscle except for aceclidine, because that's the only one that is selective just for the iris sphincter. Risk of retinal detachment is definitely there with Vuity and Colosi, as those are pilocarpine-based options. There is a theoretical risk with aceclidine, but again, aceclidine, there's never been a retinal detachment reported with aceclidine, even with it being used at least 400 million doses used in Europe. And then there is a theoretical risk of retinal detachment with carbachol, not with brimonidine, but with carbachol. But that has actually also never been reported when it comes to a topical drop. There have been retinal detachments reported with carbachol use during the time of cataract surgery, but there's also a cataract surgery being done at that time, which is something to keep in mind. So now we're going to move on to side effects. So common side effects with miotics: headaches, visual dimming, installation sight pain, redness, and blurred vision. So we see that Colosi has less side effects than Vuity because Colosi is a lower concentration pilocarpine. We see that Viz, which is aceclidine, has overall a higher percentage of side effects. So we see that patients can have headaches, installation sight pain, and dimming of vision. Now, when it comes to these drops, aceclidine has the greatest chance of causing dimming of vision. Why is that the case? Because it's constricting the pupil so much more. When you constrict the pupil so much more, you are going from a normal pupil size to all of a sudden a very, very small pupil size. There will be a dimming vision effect, not just with the aceclidine, but with all of these. Anytime you constrict the pupil very quickly, there is absolutely going to be a dimming effect. It's probably just going to be more pronounced in a drop that causes more pupillary constriction. We'll talk more about that in a little bit. And then Brumacol, headache, installation sight pain, ironically conjunctival hyperemia, even though there really shouldn't be because brimonidine is supposed to constrict those blood vessels, and then blurred vision. So going back to retinal detachments have been reported with a pilocarpine-based drop used for the treatment of presbyopia. So we know that pilocarpine does increase the risk of retinal detachments, but this has actually been seen in a drop used for presbyopia. When it comes to carbachol, again, carbachol was not used a lot topically. It was mostly used intraoperatively during cataract surgery. So I could not find a single case report of carbachol use topically that caused a retinal detachment, but you can find case reports of carbachol associated with retinal detachment when used during a surgery. It's hard to say how much of that is due to carbachol and how much of that is due to the surgery. In this one case report, what we see is that at the end of the surgery, carbachol was injected into the anterior chamber. Unexpectedly, the anterior chamber became shallow, probably because that lens became displaced anteriorly. So it caused that shallow anterior chamber. So then the surgeon ended up putting atropine in the eye to try to reverse the effects of carbachol. And then the patient ended up with a large serous retinal detachment. But again, that's also during surgery. So something to keep in mind. But if you look at drugs.com, what you'll see is that for carbachol, they do list retinal detachment as a side effect on drugs.com. So something to be aware of. Now, what about dimming and what about driving at night? This is always concerns that people have when it comes to these drops. When we constrict the pupil quite a bit, there's absolutely going to be a dimming effect initially. Is this a concern? Well, your retina is smart. The photoreceptors know how to dark adapt. So when you get a quick miotic effect, everything's going to look dark at first. And then over the course of probably the next 45 minutes to a few hours, your retina is going to dark adapt a bit and it's going to get used to less light entering the eye. And the patient's probably not going to experience much of a dimming effect at that point. There has been one study that's looked at night driving, though. So this was a study that looked at 1.25% pilocarpine. They instilled the drop, and then 1 hour after installation, they looked at night driving. And they found that patients did not perceive that they drove any worse, according to the questionnaires, but 3 of the 7 driving tasks favored the placebo. So they technically drove a little bit worse. So I guess this study is hard to glean information from because they instilled this drop right before they went and drove. They instilled the drop 1 hour later, they started driving. So yes, it is conceivable that if you constrict the pupil quite a bit and you have not had a chance to dark adapt fully, that it could affect your ability to drive. So this does support class labeling about caution with night driving, but honestly, we just need to tell our patients, don't put this drop in at nighttime right before you're about to drive. Instead, put it in the morning, and you will probably be fine because your pupils are going to be used to this. Your retina is going to be used to it by the time nighttime comes around. Should we be concerned with dimming? So I see a lot of people making all these comments like, I'm not going to prescribe a miotic because I don't want to be the person that causes a car accident. To me, that's a little bit silly because we've been using pilocarpine at high concentrations and aceclidine at high concentrations for decades, millions and millions and millions of doses, higher concentrations up to 4 times per day. And you can't find any case reports about this causing car accidents or serious functional impairments. So the people that are saying, no, I'm not going to prescribe a miotic because I'm going to cause my patients to get into a car wreck. I mean, thank God that glaucoma specialists 20 years ago didn't have that mindset. Otherwise, patients wouldn't have had controlled glaucoma because they were using a lot higher concentration pilocarpine and aceclidine to control the patient's glaucoma at that time. So I think that concern is a little bit overstated in my personal opinion. I think dimming is real, but it's probably going to be mild and transient. Patients are probably going to get used to it and dark adapt. What about diffraction? Diffraction describes the phenomenon where light waves are going to bend or spread out as they pass through a narrow opening, such as the pupil. So it really depends on illumination. So in high illumination, small pupils generate higher image quality and better focus in general. In low illumination, when you start to get to pupils that are 1 to 1.5 millimeters, the peak image quality does worsen. So, but that's roughly, you have to be under 1.5 millimeters for diffraction to become a problem in low illumination. In general, diffractive blur becomes a problem at definitely at 1 millimeter, but there's other studies that say that the best visual acuity is at 1.3 millimeters. So I think diffraction is a concern, but I honestly don't think any of these miotic drops probably get the pupil to such a small size that diffraction actually becomes a problem. I think if we use higher concentrations, it probably would be a problem. But if you even look at like the surgeries that were done on the cornea, like corneal camera inlays, they were still using a 1.6 millimeter aperture size. And that was a surgical implant that was being done into the eye. And they weren't really that concerned about diffraction with those cases. So I think diffraction's probably not a big problem with these drops. Now, headaches, you can get headaches if you have ciliary muscle spasms, such as with pilocarpine or carbachol. Interestingly, Viz did cause, or aceclidine did cause 11% of patients to have headaches. It's very unclear why those patients got headaches because we are not stimulating the ciliary muscle. Very difficult to say. In general, though, when we look at these side effects for all of these drugs, pretty much all of them are mild in nature and resolved after several days. Usually it was not a continued problem throughout the study. So that's something to keep in mind too. Pretty much all were mild in nature. Blurred vision can happen if the drug is a non-selective cholinergic agonist due to that myopic shift. Now, we're going to briefly talk about some presbyopia options that we have before closing out. So in the future, we're going to have lens softening agents. This is a drug that is going to target that lens. It's going to soften the lens. The lens is not going to be hard any longer, and we can then have the ciliary muscle end up bending the lens to allow you to accommodate at any age. The challenge is if you soften that lens too much, it could literally fall out into the vitreous. And we know that this is the case because it's happened in animal studies. There's surgeries called laser scleral microperforation, where you actually create these micropores in the sclera near the ciliary body. This increases flexibility. This allows the ciliary muscle to contract better to allow the lens to accommodate. This does show promise. This is being done mostly in Latin America and South America. Femtosecond laser for photodisruption. These are laser pulses that soften the crystalline lens, supposed to restore accommodation. It's very, very early in research stages. And then lastly, laser blended vision. This is like LASIK, but you customize the LASIK or PRK to allow you to see both far and up close. I'm personally not really sure how realistic this is, but it is being tried at this point. All right, so clinical application and patient counseling, we have to assess suitability in patients. We have to know their refractive history, their retinal status, lifestyle needs. We have to set expectations. These miotic drops, while they're great, they can reduce dependency on reading glasses. It is not going to restore vision to that when you're 20 or 30 years old. So we have to set expectations for them. You may still need supplemental spectacles at certain times or after a certain amount of time. And there may be some limitations in dim lights or prolonged reading. It's not the perfect option, but it is an option for our patients. And then, so we may have to do combination approaches too. So things like you use one of these drops after a certain amount of time, you use reading glasses. So when we are thinking about prescribing this to patients, we have to keep in mind efficacy. Which of these drops do we feel like is efficacious? What's going to work best for our patients? We have to keep in mind their visual needs, their daily activities, work demands, their desired dosing frequency. Do they really want a drop that they only have to use once a day? Are they okay with twice a day dosing? Do we want to use a preservative-free drop or a preserved drop? Most are preservative-free, but one is preserved. We have to think about risk of adverse effects. If they have a history of high myopia, of retinal detachments, things like that, if they've had LASIK and you know that they were highly myopic prior to that, we should take that into account because we know that all of these drops theoretically could cause a retinal detachment. Even if they have not been associated with a retinal detachment, the theoretical risk is there. And if you read the package insert for these drops, it will be mentioned that that is a theoretical risk. And then we have to educate our patients that they will have dimming of vision initially. Generally, that resolves over time. You dark adapt, you get used to it. It could cause headaches. A lot of these drops do cause installation site pain. But in summary, patients really want a non-surgical and non-refractive lens option for presbyopia. And finally, we actually have therapeutic options. We have pilocarpine-based drops. We have an aceclidine-based drop that is soon to be on the market. And then probably next year, we're going to have a carbachol/brimonidine-based drops. And I put this here in this bullet point. We owe it to our patients to at least mention this as an available treatment. Even if you are not sold on miotics for presbyopia. I understand it's not the perfect option out there, but it is an option for our patients. And we at least owe it to our patients to mention it to them because a lot of our patients really want a drop option for their presbyopia. You can mention the options to them. You can say, here are the pros, here are the cons, here are the different prescription options that we have out there. But I think what's going to happen to a lot of optometrists is some optometrists and ophthalmologists are not going to buy into these drops. They're not going to mention it to their patients. And all of a sudden, your patient's going to go talk to one of their friends, and their friend is going to have been prescribed one of these drops. And the friend is going to say, oh, yeah, I was prescribed this drop by my optometrist to reduce my dependency on reading glasses. Your optometrist did mention this to you. And I think it's going to reflect poorly on some of the optometrists that are not at least mentioning it to their patients. So I do think that it is something that we should discuss with our patients. We also need to be educated on the risks, side effects, and the pros and cons of each option. And then in the back of our minds, we need to know that in coming decades, there's also going to be more options, that miotics are the current drop option that we have for presbyopia. But in the future, there is likely going to be other options